Hello, everybody, and welcome to a special live stream at the behest of Mr. Chris Snyper, who won the topic selection for a hour and a half live stream in one of the past auctions, and here we are. We're doing it. I'm just going to start things off with a little bit of an update for everybody. Say hi to who's here. Got a couple people here. We'll say hi to shortly. So the last 2 months, I don't know if I mentioned, Angel and I got finally fully triple layers married and all that stuff. So that happened last month, December 21st. So that happened. Again, I can't remember if I've streamed anything since then, uh, update wise. Anyways, so that happened, and things have been really good lately with a mixture of stuff. I'm having car issues again. Thinking about selling my car, getting it fixed, selling it, trying to balance that with the uh, trip coming up and some other stuff, and also having to drive out to Holly to the other place all the time. Some major decisions needing to be made. Sucks. Added on the pile. But the fencing is done in Holly. We're just sort of waiting for they have to put some truck gates on. There was some manufacturing problem with the first batch they brought, so we're waiting for them to get the second batch in, which was supposed to be by the end of this month. So we'll see if that happens. If that happens, then the paddock behind the barn is completely done, and the dogs and goats will be moving out there ASAP, and then me as well. So I'm still here in Livonia. And basically, what I've been doing was I was supposed to take December to do site work. Then I take December to do site work. I made a tactical decision to basically redo every bug enclosure in the basement bug room. So I do this piecemeal because I do this every month, and most stuff in here only needs to be touched up on every month. And so I haven't gone through and done that here. A couple things I have, not everything I'm noticing is falling. Anyways, so the importance of line tracking is the label is falling off. I found a fix for that actually. I'm just using packing tape to adhere the labels now. He's using these little plastic or something boogers, whatever they're called. They're little adhesive tacky things. Um, Packing tape has been working just fine. It's not leaving a lot of residue, etc. So we're also looking at the building is going to take about 6 weeks from start to finish, and it's should start groundbreaking basically as soon as the ground thaws. Now with the paddock and all that stuff done, the land clearing done, that crew can get in there and they can start as soon as the ground thaws, and then 6-ish weeks later begins the grand move, and then fixing up my house here for sale and all this other stuff. So things are about to get busy as soon as we're just waiting for spring. So I made a tactical decision to, instead of working on the site, which, if I don't have bugs to sell, it's kind of silly having the site, blah, blah, blah. Investing time in redoing all of the basement enclosures, which had not been really seriously touched on since maybe 2022, possibly 2021. So just going shelf by shelf, everything that needed a deep overhaul, completely gutting it. It's a bunch of 10-gallon, 5.5-gallon tanks that have to be cleaned out and restaged, and new substrate and all this stuff. But it's going to cause a lot of the cultures to have some pretty big booms and be ready for the spring auction, which is at the end of April, and ultimately for the move to get us ready for the next phase of Roach Crossing. So with my time being limited, basically as soon as spring starts, I figured it was a better idea to get everything set up so I can be a little bit more negligent while I have other things to tend to before the move. So for website stuff, Chris and I did do some communication on that. And I have some time scheduled in February once I'm done with the basement work to basically 3 days straight. I'm going over my friend who works for the government's house, who says he's behind on some work that he needs to do, and we're gonna do a 3-day nothing but work on stuff. And I'm pretty sure I'm going to be able to get a chunk of what I need to get done. Then I have been adding to the list actually, because every time when I make a label, it basically says, okay, I've got this figured out. It's a permanent part of the collection. So everything gets an identification number. Hey, this segues into what we're doing today. I'll just pop this one off the Aaron Vega patch here. Everything gets, so you have the Latin name, and I may touch on how I'm like labeling those sort of things. Latin name, the locality, the strain or cultivar information. When I acquired that specific group of them, so some of these species I worked with for much longer, but if I lose a culture, then I use the acquisition date is the restart date on the culture. And then the RCID, which all the roaches are just going to have numbers. And then for everything else, like general insects, isopods, there's currently 2 other categories, isopods, and then basically all other invertebrates. There will be probably ISO, and then number will be the RCID number for different isopods, and then probably bug, and then a number will be that. So everything will have a 1 through however many numbers on their RCID. And the purpose of this is that that number will never change. So we could find out, oh, actually, this is the locality. It was actually from this other place. And, oh, actually, it's not. This happened very recently with Argola pilosa. Oh, it's actually not pilosa. We don't know what it is. It could be Sylphoides, but we're not 100% sure. So, so the RCID will never change. So if somebody goes to my site and they're like, what? I bought this as this other species. It's this now? The RCID will be unchanging. And this is part of the system going forward. So anyways, I have been adding some new stuff to the list, almost at 300 different types of roaches. I'm pretty sure if I added some stranglers that are in here, we might just be tickling 300, which is wonderful, and it will be a big landmark. So anyway, so that's just some general updates with everybody. The cold has thrown a wrench into shipping, including the FedEx shipping. It's just been miserable. A blessing in disguise, because it gives me a good excuse to focus on doing husbandry and not get worked up to do all the shipping stuff that I usually do. So that's how things are going. I'll take this time now to say hello to everybody. Courtney's here talking about a strain called Cherry Punch of something. Don't know what. Also, I just realized I forgot. Either I've hit the number of subscribers or the number of viewers again or something, but YouTube has turned back on my, like, what's this, goal? Make a super chat, more fun with a goal. Choose the super chat color, set timers. Hey, yeah, I could turn that on. So they've turned back on whatever this monetized content type stuff is. I don't know if I want to play around with it today. We won't have the normal intensity and diversity of viewership, but maybe that'd be a good thing to play around with the next auction or something. So anyways, Aquarimax Pets says, Kyle, in the years I've been following your website and YouTube channel, you've certainly helped me better understand the importance of line tracking in invertebrates. Yes. Thank you for being in the same camp. Hyperbeam says, 100% agree with the statement above. You helped me understand how worthless and bastardized my magic potions were, and I haven't been able to love them ever since. We've got Duke and Enzo, the Iggy's are here. Alan's here. Exotic Wilderness. Joseph Tavir. Everybody asked about how my Esculca are doing. I've got Pupae sitting right over there, and then I think the rest of them are somewhere on the high shelf up there, waiting to hopefully a close, and we'll see how the adults are doing and all that stuff. Is that a top loader for trading cards? Yes, from Hyperbeam. Yes, it is. It's a solid top loader. And that way I can, if I need to update a label or stuff gets gross and nasty and needs to be redone, if there's a typo or something, I can just slip out the thing and then push it back in. And I really love this. It would be hard to beat this system, I think, for labeling things right now, especially guaranteeing that the things don't stick off with the tape. Duke and Enzo saying that this is ingenious. Yes, I don't praise me too much. Don't let it go to my head. That's an interesting way of labeling culture, says Exotic Wilderness. It's really good for. I want everything to end up in a culture, in a container big enough that they can have one of those labels on it. It may not happen for some things, but it's the goal. 13 viewers and 13 likes, says Duke and Enzo the Iggies. Yes, please give me some likies. John Doe's here, LC's here, Braden's here. I think I saw Behijager. Yes, Behijager's here too. Altered Pants, we got a lot of regulars who I bet you guys are aching and missing the wiggle, and so am I. It's really my weeks have been a lot different without the wiggle, but boys, is it really the time to sow and not the time to reap right now? Adam Hawks, hello Hawks, Adam Hawks, Adam. Good to see you. Victor's barking. I think Angela's here. She's supposed to come over tonight. We have a big day planned tomorrow to kind of get through some of the stress. Yes, Duke and Enzo talk about the snow and ice in South Carolina. Yeah, it's been something. We were in the single digits, and that's always stressful around here. Of course, the dogs didn't care. They wanted to be outside most of the time. So, but let's get into the topic of the night, which is basically strain tracking, etc. We have a beautiful thumbnail pictures provided by Martin, and I don't know how to say your last name, Marvin, Mr. Vic Mervic. The Wild Martin saw my terrible images of pseudoglomerus. I snapped in a couple of minutes just to get something on the thumbnail, and said, "Oh, hey, you can use my beautiful macro photography images of Pseudoglomerus magnifica of 3 different strains," and there you go. So Duke and Enzo asked about my Armadillidium badium. They were Casteldacia is where mine are from. Anyway, so yeah, Angel's definitely here because now Nazi is starting to get worked up. So I apologize for any background noises. So let me just go through. I think I had a very brief list of what I wanted to talk about today in terms of an order of things. History of captive bugs and the consequences of carelessness. All right, so let's just go into things. Let me just talk about where things were when I got into roaches specifically, because I work mostly with roaches and I guess isopods too. But getting into roaches and just kind of looking at the trajectory of things from there is a lot of the stuff just was not tracked. That's just the bottom line. We have a lot of roaches from the very beginning of propagation that just were never tracked where they came from. We have had dubious species IDs on things. Trying to think of one that's been changed. It's even for example, it's even possible that Blaptica dubia is not actually our stock is not actually Blaptica dubia. So you've got that. So there's a combination of some things here, and this is just kind of how stuff goes with. I'd say, oh, we did bad. Everybody else is doing better. It's just kind of how a lot of stuff has always been. People collect plants. They don't. They get into horticulture, or just thousands of years ago, stuff has been collected. We're like, well, it may have originated from this area, but it's been hybridized with this, and this so many times we don't exactly have a point of origin. Somebody posted in the Discord the other day that the captive population of axolotls may actually be hybrids with a closely related species from when they were originally collected, whatever that was, early 1900s or something, for laboratory research. They may have mixed together other species that were collected and sold at the market. So this stuff happens. It's just kind of commonplace in the domestication process with a lot of organisms. I mean, there's even my precious the gompers. There's introgression of even wolf genetics in there from hundreds of years ago, not even thousands of years ago, hundreds of years ago. The introgression of different species into a captive animal. Pigs, they will breed with wild boars, and you get all that stuff. It gets even weirder. You look into stuff like cows and buffalo, bison in North America, and they will hybridize, and you end up with their herds of bison in the U.S. that have a lot of cattle genetics in them. So So monkey bring up domestic chinchillas are almost certainly a hybrid between 2 or more wild species. So, you know, this stuff happens, and it's just part and parcel of the process of domestication. So there's value in it for sometimes things hybridize, and there's a fitness curve of basically there's the curve is kind of goes like this. It's kind of surprising in terms of the effects of inbreeding to outbreeding. You kind of start over here with inbreeding. You got too much inbreeding, and you have kind of a fitness and overall fitness detraction, and then you have this sort of sweet spot of outbreeding or outbreeding and inbreeding where there's this sort of all around good fitness, and then you kind of had this like really drastic drop off in fitness in terms of the more unrelated two organisms are, and that's called outbreeding depression. A good example of this is mules. So horses and donkeys, they're both genus Equus, and they can hybridize. And you know, you have people saying, "Oh yeah, outbreeding, it's great, it's amazing. I got to outbreed my everything." You outbreed a horse to a donkey, yeah, they're relatives, right? You would say, "Oh, if I bred them, I get all these strong characteristics, etc." And mules do have certain characteristics that are desirable, but they're genetically incompatible, and so you have the ultimate result of this incompatibility, which is sterilization. So there's just no perpetuation of those genetics past that point. So you have this just ultimate lack of fitness in terms of infertility. That population consisting of mules cannot reproduce. It does not continue on into the future. And then you have extreme inbreeding, where you have, you know, in human cases, you have a lot of recessive traits pop up, and then you have issues from that where, yeah, these people who have these deleterious alleles, they can still reproduce and they can still produce fertile offspring, but there are social effects, there are mental effects, etc. With that situation. So you've got kind of these two extremes. But again, a thing to note here is the idea that it is, you know, if you're going from a strictly evolutionary side, but we're not talking about morality or ethics or spirituality or any of that other stuff. From a strictly evolutionary standpoint, inbreeding is far more favorable. The extreme of inbreeding is far more favorable than the extreme of outbreeding, because you end up with sterility and outbreeding cases. You have species that are hermaphroditic, that are self-hermaphrodites, where that's like the most extreme form of inbreeding you can have, where you're mating with yourself, and there are some species that will perpetuate almost entirely by those means. So there's a lot of discussion here that I'll poke into in a minute here with some stuff. So this factors into something with captive insects, is this idea of this reduction of fitness in captive populations that then encourages you, or theoretically encourages the keeper to outbreed. A lot of people are like, oh yeah, I really need to, my dubia started dying off and I really need to like outbreed them. I think they're getting too inbred. And it's like, that's not how that works. So that's kind of where things were earlier on. People were just excited to have different species of cockroaches, which is great, and we still have many of those strains to this day. Never have been added to with wild stock. There was some ridiculous Facebook post, maybe the peak of Facebook bug insanity. I think Alan linked it to me, where somebody was talking about, like, somebody asked, just a question. Somebody asked a question like, is like inbreeding bad for like captive bugs, etc.? And you had some people just like swoop in from out of nowhere with these crazy things. Like this one guy was like, yes, captive invertebrate breeders, they go out and they wild collect new stock every couple of years to prevent inbreeding. And it's like, literally, who are you talking about? Actually, I want to know who they are, who is so crazy and somehow has the means to go out and wild collect roaches to prevent inbreeding, or just anything provides a counterexample. Like, who is going out to wild collect more Simondoa? You can't. Is the bottom line. So yeah, so originally people were just happy to have different species. There's this idea of, oh, you gotta like outbreed them, etc. There was also a lot of, it seemed like there may have been some carelessness because it was just, and surprisingly, this didn't really seem to start until I got into roaches. There was some golden period of roach keeping before I got into things where things were kind of very fresh, other than stuff like hissers and discoids that had been propagated in labs for a long time. So people were getting the things were coming in, and people were keeping everything separate, etc. And then right around 2011, 2012, when there was a bigger push for diversity of feeder roaches, is where sort of some of these problems started materializing. I take that back. I almost forget because they're not around anymore. We had Glabrous cranifer hybrids when I got in. People were labeling them. They were a cross between whatever C. peruvianus or Fusca are and cranifer, and they were not vigorous at all. I tried to keep them a couple of times for, oh, it's another roach to have, in the past, and they just never did well. They had all kinds of wing molting, deformities, etc. And this is an example of that high the outbreeding depression, and also a good case for why cranifer and whatever Fusca or C. peruvianus are different species. Even if they were synonymized both as Glabrous cranifer, they need to be split again because they do not produce fertile hybrids, or there is such a dramatic lack of vigor. It's like, yeah, there's some incompatibility here. So, but anyways, so just to add to emphasize, you do not see that line of roach available anymore. I don't think I've even heard of them since maybe 2012 or 2013. They were just this thing that happened. Somebody mixed colonies on a whim or accidentally, whatever, and they created this hybrid stock, and they at least had the sense to label it, and they just naturally died out because they had too many issues. They had to be very accommodated for, and people just did not do that, and they died out. Another one that 2012ish was when the Blabrus discordalis and boliviensis started getting mixed together. There were a couple of large roach breeders, one of whom is still around, who would just do the, oh yes, I have to outbreed. I like to mix up the genetics, which is fine. That's part of that domestication process. But when you don't track what's going into something and then you sell it as if it's the pure something, now you're sending hybrids all over the place. You're compromising pure stock. You're jeopardizing the fitness of people's colonies. You are potentially breaking laws. That's a gray area with some stuff, but in the sense that if you are selling discoids to Florida, and nowadays there's a big Canadian market to Florida or to Canada, and they are compromised with Boliviensis genetics, you are no longer selling Blabrus discordalis. You are now selling hybrids, which are illegal. So there's stuff like that. Then there's also the captive conservation of the stock perspective of things too. So with the Boliviensis, uh, they were more common back in the day when I got into roaches. They were just sort of ramping up in popularity, and then around maybe 2015, 2016, they really fell off for whatever reason. And now I think I'd be hard pressed to think of somebody else in the United States who has Boliviensis. Alan, you can correct me if I'm incorrect, if you're here, but I might be one of the only people who consistently maintains a colony of Blabrus boliviensis. So, so moving on, the importance of all this stuff is I'm going to dig into the commercial aspect of it. I'm going to talk more about the moral, ethical, whatever part of it right now. So with that, the bottom line is most stuff, just having that information is important for a number of reasons. Um, you in case there is a change of identification, in case, oh, I got the stock back in the day. Oh, those aren't called that anymore. That's this other thing. Oh, okay. So that helps with with sharing information about husbandry and just labeling stock. You know, people see a picture of something from years ago, say, "Oh, I want that. I'm interested. I read about this. I want to work with it. I want to see. Oh, it does this one thing. I would like to see that. I'd like to. That might be useful for something else I'm doing, like a feeder or handling or something. There's something I can use it for." And then you get the stock, and it's nothing like what you had read about. And then it's whiplash of, oh, well, who's in the right, who's in the wrong, what happened, what changed, etc. Um, it's important for keeping track of when you're doing crosses of things, so there's some degree of replicatability of, oh, maybe I crossed this isopod strain with this other strain and I got this result, and that was because of the unique histories of both of those strains. A good example of this was just yesterday. I think Alan sent me a picture of some breeder in Europe who had posted, oh, I have a new Armadillidium nisatum variety going, and here's these beautiful orange Dalmatian nisatum with big, bright orange spots, etc. And I looked at it as like, Alan, that's like, you know, that must be either that's not nisatum or that must not be like in the U.S. because I've done that cross before, and you don't get those consistent, beautiful results. There's just too many background differences in the U.S. stock of nisatum. The Dalmatians don't present that way. The orange Dalmatians don't present that way. The regular Dalmatians barely present that way, actually. And Alan said, yes, this person's in Europe. So both of those stocks people might just be inclined to just call them orange Dalmatian. And there's a big difference between the two of them in the way they present. So now I see this picture and I say, oh, I love orange nisatum orange Dalmatian. I'm a person who really cares about the PPQ permits, all this other stuff, or I'm in Florida and I want to stick to the rules, blah, blah, blah. I want to get Armadillidium nisatum Dalmatian. It's one of the only things I can have, whatever. And then you order them and you get the U.S. orange Dalmatian stock, and then you look at them and you think, I got scammed. These don't look anything like those pictures. What's going on? The seller sucks. I was lied to. You know, what's going on here? Not knowing that there was a difference between the two lines there. That's another part of it. And then getting into the conservation aspect of things. And this is a weird example because it's not. This is not a conservation in the, yes, we must preserve the species. The Simondoa are extinct now, and now it's our job to preserve them until one day they can be released in the wild. This is kind of a weirder side of this. Blaberus craniifer, there's a fair chance they're extinct in Key West. I've been there personally several times looking for them. I know people are looking for them. I know people who have put out bounties for them. We've been to spots where they were reported not even 10 years ago, something like that, and they're not present, etc. So assuming we had collected some stock that was labeled with that information, that's a part of that species' natural history that is then preserved. We can say this stock came from Key West. It's extirpated there, but it's from there. And maybe when that population was split off there, it had certain characteristics that are not as frequent in its source population, that went extinct in its source population. There are different things that come and go in populations, and when something gets isolated like that, it's a good chance for weird stuff to pop up. And so now we don't have any stock from there. Even if we did, we don't know because nobody labeled it and all of this other stuff. And a case in point for this, you have a trait that pops up that may not be present elsewhere, is the Caracolus marginella populations in Florida. There's one population that is all just the regular spiral, the yellow and black or brownish spiral on the shell. And there's another population where I think Alan or Satchel or Kai reported that it's something like most of the individuals, I don't know, Alan, would you say 80%, 90%, are, instead of having that spiral on them, are just a solid burgundy color, very attractive burgundy color. And that could be because those two populations were from different sources. And it could also be at the other extreme end of things that, that site has Caracolus marginella, and it also has another species that's currently being misidentified as Caracolus marginella. So there's a lot of that stuff going on too. So if we collect that, we don't label it, we don't know where it came from, we don't know its history, we don't know what there's no idea of standard for traits or other things that it could present. and that information is very easily lost to the ages. And along with it, a lot of the value of its story and this other information. So, anyways, I'm going to take a moment here to just go up through this. We have some Circa bring up there's some information here about uh, the fact that the axolotls are fairly likely to be captive axolotls are fairly likely to be hybrids between two species. And if I recall, some of that was done. Yeah, Circa's pointing out here during the 1900s, the AGSC population saw multiple introductions of outside axolotls to uncover new recessive mutant alleles for developmental research. So again, they're getting stock from outside. They're crossing with domestic stock, assuming they're both the same species in search of these new alleles, and they end up making a hybrid strain, which is again very par for the course for domestication and domesticating an animal. So. The axolotl controversy goes deeper than I thought. Very interesting, says Hyperbeam. We got some discussion of penis fencing and flatworms from Duke and Enzo the Iggies. Insect Central asking, what's the difference between a parthenogenetic species versus one that species sexually reproduces? So, parthenogenesis and self-fertilization are two different things. Parthenogenesis is cloning of self, and usually involves something like an unfertilized egg dividing within there's like dozens of different ways that this can happen, the parthenogenesis, but the resulting offspring are genetic clones of the parent, and there can still be mutations and things like that, but it's not as they are most of the time they are clones of the individual that produced them. Self-fertilization is where the individual possesses both male and female parts and reproduces sexually with itself. So there's a sperm and an egg, or there's weird variations on this too. But you can end up with offspring that are very different from the parents, because even if they had if they carried one allele for a simple recessive trait and then they self-fertilize, you end up with um, brains stop. It was a Punnett square of offspring, so you end up with the homozygous dominant and then the heterozygous and the homozygous recessive individuals. So you actually end up with in self-fertilization versus parthenogenesis, you can end up with a good selection of diversity of characteristics in the offspring, versus parthenogenesis, they're all direct clones of the parent with minor mutations, and it sometimes can be number of chromosomes can vary. Sometimes it's there can be mutations at a single locus that do weird things, etc. So that's the difference between those. Circa bringing up that Circa saying that the axolotl situation is because of poor breeding practices. They have about 5% of tiger salamander DNA from the GMO albino ones. So, also Stephen Logan, yes, we are back. Courtney blaming Elon for all of this. Alan says he doesn't know anyone who has Blaberus boliviensis anymore. Duke and Enzo saying never heard of Orange Dalmatian Nizatum. I have not released them because the U.S. strain of Nizatum, the Dalmatian strain, it's been called Pearl. It was isolated by Valerie O'Neill back in like 2017 or something like that. She called them Pearl because under the conditions she was raising them in, they just looked like they were white, almost like a leucistic or a whiteout. But they're actually Dalmatians. So when you keep them drier and cooler, that trait expresses more. You can actually see that they're not leucistics or whiteouts. They're actually Dalmatians. So crossing that to orange or peach or any of the different orange lines of Nazatum, you then produce hets and hets for both characteristics. And you basically, it's very difficult to tell if a Nazatum is both Dalmatian and orange at the same time, if it has both those recessive alleles, because having just one of them lightens the body color, and then obviously if they're homozygous recessive for one of the traits, you can tell, but it's difficult because when the spots don't show, it's very difficult to see if they're orange. So I've been manipulating a group. I've been keeping them cold and dry to get as much expression of the spots as possible so that I can then go in and say, ah, yes, here's a male with orange spots. Ah, yes, here's a female with orange spots. Then breed them together, and then you can be sure that all the offspring are going to be orange Dalmatians. It's been hard, and I have a couple of individuals separated, and what I might actually do, because I can see that they're Dalmatians, but I'm not 100% sure that they're homozygous recessive for orange. They might be het, because het for orange Nazatum do have an orangish or in the males purplish color, so it's very hard to see. So what I'm thinking I'm going to do is I'm going to keep those individuals isolated and then cross them to orange Nazatum, and that way the offspring will be they are guaranteed to be mostly orange, and then any that are the whitish color, the Dalmatian, I can pick out from there. But it's a very meticulous process, and most people do not do this single pairing etc. stuff for them. They will throw a bunch together, and when they get most of the individuals looking like what they want, they will just say, okay, it's isolated. And that's how you get situations with stuff going out that's incompletely separated. One of the most recent examples with this is the Cubaris species Panda King, red panda, which still throw a lot of the solid oranges, or maybe it's vice versa. One of the lines was called citrus, one was called red panda, and one of them throws the other one. I'm pretty sure it's the white and orange ones still throw solids, and that's from poor isolation of the characteristics. Circus says, I wonder if it's even possible to release a lot of hobby stock into the wild. Very unethical. Releasing up, and other people agreeing with their introducing threats to the native ecosystem, blah, blah, blah. Yeah, they tried to back cross on this about the XLs, and apparently historical XL lines were lost at the facility in 2000s, according to that paper. I wonder if a black light would help you isolate his aim. I might try that and see if that shows any of the colors in a different light. Now Angel's definitely here because the dogs are going crazy. So anyways, so that's that situation with the Nazatum, and again, I can't really remember exactly where we were on this. I mean, I will just go for hours on this topic, but I can't remember where we were at before the Nazatum. Oh, I was just reading through these comments, I think, and I was going to get into the next level of things, which is kind of how things are in other hobbies. So with the reptile hobby, which is the closest thing adjacent to the invertebrate hobby, and definitely the invertebrate hobby has been buddy with that sort of community for a long time. Before all of this was basically the fish keeping community, where they would kind of keep track of stuff, and stuff would only come from so many places. So, we got a shipment from the border of Myanmar and Burma, and then people would be like, oh yeah. And when every river and every tributary has its own species in it, it's kind of easier. It's like, oh yes, the species only found here, so that's where the stock came from. Or we're going to describe the stock that just came in, and it came from this location and maybe present in nearby tributaries, etc. But the fish keeping hobby, they do keep track of some things, and with all of this stuff, it's you will pay more for locality stock, things that have this value of, yes, this is what I'm saying it is, and you're not going to get any surprises. With reptile keeping, there's that too. People keep track of all the gray-banded kings. People keep track of, what's another thing, the rosy boa localities. People cross stuff, they make new things. People hybridize all the corn snakes, the rat snakes, the gopher snakes, the milk snakes, the king snakes. They've been making these crazy 8-way hybrids of all these different colubrid snakes, and they track the percentage of it, thankfully. But with reptiles, you can really see when you go from point A to point B and the same species is present in both places that there can be differences between those populations. And so with insects, it can be difficult about where to draw lines. With invertebrates, it can be difficult to draw lines. Um, so things fly, they travel very far. Uh, there are, you wouldn't say that the monarch population in Michigan is genetically distinct from the ones in Kansas on some large degree. And yet, despite the fact that monarchs fly thousands of miles every summer, there are resident and genetically distinct colonies of them in California, in Florida, I think Madagascar they're introduced to, Hawaii as well. And those populations have rapidly diverged in terms of their genetic composition and then the expression of behaviors and different other traits in a relatively short amount of time. So I don't know if you were looking from a conservation perspective, if you would say, okay, the mass monarch migrations of the eastern United States are over. The population went extinct in Mexico, but everybody cared so much that we ended up buying 1,000. No, you probably need more than that. 20,000. Let's be ridiculous. 1,000,000-acre preserve for the monarchs and the dead center of their old overwintering grounds before they went extinct. And now we will reintroduce them using a population from Hawaii because they're all monarchs. I don't think that they would do that. They would probably look for semi-migratory populations that are still in Florida or California or something like that, and try to facilitate the reemergence of these genes that may be present in the more closely related populations that may have had some genetic influx from the migratory population, etc., for the conservation perspective. So, um, Elsie says, I have Blabber's Brilliances. I love them. Sad not many people have them. I'm glad to hear that you have them. Hold on to them. Hold on to them dearly and tightly. So that's the value of some things. There's a particular individual who years ago infamously was saying that he could not see the difference between two different localities of ivory millipedes, and therefore they must be the same. I could see that logic in the sense that, okay, you have a person who came to you and said, oh, I just bought some ivory millipedes from the pet store. Do these look like they're a different thing than what you have? And maybe having some knowledge or information that, oh, these came from a pet store. There's only like one guy in the United States who wild collects ivory millipedes for the commercial market to just resell. There's a good chance that this is the same as what you have, that it's the same, that this is going around. So I could see that, although I highly doubt that's exactly that's what went down in any capacity. But to just look at two things, there's so much wrong with that, and it assumes that you know a great deal more than you do about that organism. Uh, you're looking at a slice of, maybe they're only bringing you a couple of what they brought. Maybe they only have a few. You are looking at not a representative sample of the population of this captive stock this person has. They may not be adults. You may look at them and say, oh yeah, those are from here, and not realize that what they just showed you are actually immatures. You don't know how long they've had them for. There's so much information. You don't know how their husbandry was. They could have kept them too dry and now they look darker, or really moist and now they look lighter, etc. So it's best to err on the side of saying I don't know, and to be sure you should not do more than that with things. Zen Monkey bringing up that he can't tell the difference between a coral snake and a milk snake, therefore they are the same. That's kind of like the logic going into things here. Another example, what's an example of two things that are pretty much identical, that two invertebrates are pretty much identical. There's a lot of snails. I'll fall back onto that one. There are a lot of snails that somebody without a passion for snails would look at and say these are the same animal. And there are I think somebody brought up in the server the other day, there are some things like the those weird worms that I found in my uh, moth fly enclosure, where he's like, oh yeah, even the guy who did the taxonomy on those is like, I don't know what those are. Brachypelma hybrid situation, Alan bringing that up too. Again, it just happens all over the place, and a lot of us sit and we hope that one day our stock would be used for what I would say is maybe one of the greatest goods of introducing it back into the wild somewhere where it was removed from. We can all hope that that could happen. Um, that's not usually the case. That's an extreme rarity. Um, so, but there's a lot of bugs in the U.S. that have a possibility of going extinct, and not all of them are going to get federal funding, and not all of them are going to be charismatic, and all this other stuff like the American burying beetle. It's propped up by the government, and then they started this release program, and now I think there might be some functional wild populations, etc. And that's great because it's a large charismatic species. It's colorful. It's people are interested in morbid things, especially in the last 20 years more than ever before. It's cool. It's weird. It eats dead stuff. I'm so quirky and original. I like death and blah blah blah. Um, but then there's also ecologically functional, removing dead things from the wild, which we always appreciate something that does that when there's a human health risk. But you know, there's a ton of crayfish in Alabama. There's a ton of snails. There's stuff all over. I didn't even realize. I don't know why I looked it up to see what species were endemic to North Carolina. There's a bunch of animals that are endemic to North Carolina. I thought, like, how? Like, what's so special about North Carolina versus like going a little far south or a little far north? Like, it seems like it's in a good place for general biodiversity, but not a space that I would assume would have a lot of endemism. And guess what? It does. And even Michigan, up until recently, we we have a couple of endemic invertebrates. There's some riffle beetle that's found in like one little tributary somewhere in the middle of the state. And there's a pseudoscorpion that was described from southeastern Michigan that's never been found anywhere else. Michigan, we were under a glacier until like 10,000 years ago or something. And then I'm forgetting the main one I was going to bring up. The tamarack tree cricket is endemic to Michigan and part of adjacent Ontario. I think they said that might be a more recent introduction or something like that. So you got stuff like that going on. You'd never think that. And so we all hope that stuff could be released back in the wild. It's very unlikely that will ever happen with any captive invertebrates for a number of reasons. And even then, if it was released, will it establish a population, and who cares, and blah blah. But if you get it going in captivity, it's something special. And if the world at large loses it and you still have it, it has immense value. And that's from our guys' perspective, from the bug keeper's perspective. That's not a shared common sentiment about a lot of things, but still. So how many species are undescribed? Many species are undescribed. People are working on them. Yeah, we're there's some undescribed roaches in the U.S. that are being worked on as we speak, and I guarantee you the greater public at large is not going to run around demanding to save the roaches or even see any sort of value in that sort of thing. But anyway, so that's one justification. I feel that's the highest good justification. But at the end of the day, it's more about being good and proper and professional with keeping these organisms as part of your responsibility to the hobby and ultimately to the world at large in doing so. so that everybody's on the same page and that we share this knowledge and we move forward with this knowledge in our future endeavors of keeping bugs, making, getting bugs out there to the general population, sharing bugs with other hobbyists, etc., etc. And I realize that this is not a, well, you gotta do all this crazy work for literally everything you get. You gotta, that giant African millipede that you just got in because they're all supposedly captive bred now, even though they're a mixture of two different species and they're coming in for relatively dirt cheap and they're getting shipped all over the world and they're coming in, they're supposedly captive bred. You got to find out exactly where those things came from, or you're a bad bug keeper. It's like you can only do so much. But my argument is, when it's easy to do that much, when it's basically harder to throw that information away than it is to keep it somewhere or to share it somewhere, then I feel you have a moral obligation to do that, and doing so shows a level of negligence or disregard. Basically, you're not a professional. You don't really care about what you're working with, and you're in it for the money or the clout or some other reason. So Alan says, believe the Procambers virginalis, I think that's the marmot crab, believed to have been produced in captivity because someone mixed two different closely related species of crayfish with different ploidy. I would argue what probably happened there with the Procamber situation, if that is the marmot crab, I think. Um, yeah, the marbled crayfish. What probably happened there was, since they originated in Germany, I think, was somebody probably received crayfish from the United States, and they may have been caught by a bait dealer or by somebody who just, this person wanted crayfish from the US. So this person collected crayfish from the US, from different tributaries in their home state, whatever. They threw them in a bucket, they kept them alive, and they just threw them all together and they sent them to somebody in Europe. And so this person would have received a mixed group of just all these different crayfish that probably didn't come from the same body of water, but they were interested in because they came from the United States and they wanted North American crayfish. And then you have an entry point for mixing things that would not normally mix, and the point of origin as well. So Duke and Enzo the Iggy's getting, here we go. Kyle, what is your opinion on collectors in Asia who purposely withhold locality information and monopolize on collection sites? So I don't blame them. With something like isopods, where you have a lot of people in the West who you probably perceive as very stupid, but with a lot of money, and so you would just catch whatever you can. You want to be tight-lipped because there are a lot of other people who see it as easy money who could go to your sites and collect there. And that's still the way it is with some stuff in the U.S. Some of my millipede stock is only labeled to county level, and I'm lucky that I even have that information, because a collector is terrified that somebody will find their spots and clean them out and make money and whatever, disrupt the market. So I understand why they're doing it. I don't like it. What is more despicable than that to me is that they are collecting things, isolating morphs, most of the time very poorly, and then labeling it as a different species, and then sending it to the US. So we then receive something that we have no idea if it's expressing, if this is the natural color, if this is the natural general phenotype for the species, if it's a solid or a mix, or it's pink, or it's this, or it's that. And then we have no idea where it came from. And even more despicable than that is taking something like these cherry blossom Cubaris species pacchongs and calling them species cherry blossom because it sells better, because people don't want to see this list of stuff on the name. It discourages them from buying. They want to just see species cherry blossom so they can just get it, and then they have it, and it's this incredible thing, and they can sell it for exactly what they paid for it. And that is just, I said, this is exactly what I said would happen years ago, back in 2018, 2019. I said, people will mix things up. And then when you allow the mixing up on purpose, When that is normalized in a community or an industry or whatever, then you open the floodgates for people to intentionally obfuscate and mislead and swindle people. And that's exactly what's happening. Now you see people have gotten cherry blossoms, and the natural color is not what the original pictures were showing. So, um, over collection, that's another thing that can happen. So here's a perspective on over collection from an older friend of mine, from Mr. Kim Wisman. So I had brought up, he was the first person who got me to think about the inbreeding situation, because I used to be, I was pretty much literally OCD with roaches. Like, I would be sitting in school, in high school, and like fretting over whether or not my discoids were going to be like compromised with another roach species or my cranifer. Like, I would sit there and I would fixate on that for like hours, and then get home and immediately check on them and make sure everything was fine. So there was actually like actual OCD about stuff like that, especially when I was younger. Now I'm more confident in my keeping methods, so that doesn't happen. So I'm not that obsessive. But I was young, and I had to figure out what I was doing. I was doing it my own way and all this other stuff. Um, but anyways, I would sometimes think about inbreeding. It never overwhelmed me enough to be like, oh yeah, I gotta mix all my roaches and get some new genetics. That never happened to me, fortunately, because I was more afraid of compromising stock than I was of inbreeding. And I'm glad about that. So Kim, at some point, I said, oh yeah, my Princessia aren't breeding. I think they're inbred. And Kim just sort of like looked at me like, no, that's not what's going on. It's like, that is not a problem in almost any captive organism. This is, we have species that have been bred in captivity for thousands of years, like silkworms, that are insanely inbred, and they don't have those problems. There's something else going on. And I was like, huh. And then I looked into it more, and my, stuff like my Elliptorina David I thought, oh, it's inbreeding. No, no, it was poor husbandry. It was a shift in husbandry caused by changes in the room between seasons that then affected what was going on in the bin. And that was a big enough difference that it affected them negatively. And the same thing had happened with my Princessia at that time. Um, so, where was I going with this? Oh, inbreeding. And then the other thing was, with over collection of stuff. So Kim was talking about, from being a collector in Arizona for 30 years and being part of communities of people who live there, um, before that, being in touch with collectors and stuff before that, he was saying, people are always concerned about, you know, oh, this butterfly population is going extinct. There's too many people collecting it. Oh, this is becoming rarer. Oh, people go all over the place with this. And people will obsess about something for a couple of years. They may, some legislation, something may be passed to discourage people from collecting or prevent, etc. And then the next thing you know, oh, the population has rebounded. And then people start collecting it again, and the population's fine, etc. And then some unreasonable amount of time later, say, oh, the population's dipping again. It must be the overcollecting. With wild invertebrates, especially those with more complex life cycles, or more complex life stages, etc. Um, things could be, you could go to a place and clean it out and still leave 90% of the population. There are things will hide under bark, deep in trees, crags, in the ground. Some could be eggs that haven't hatched for 3 years, even though the rest of the population is booming. They could just be sitting dormant for years. And you would have no idea unless you turned over every stone and cut down every tree and did all this other stuff. So, um, for some things, over collection is not the problem with stuff. Sure, if you had somebody go to a place with bugs for 50 years and they harvested everything they could see of that species for 50 whole years, yeah, maybe you'd have an argument. But he was saying that with these butterflies, everybody said, oh, the population is going up, oh, the population is going down. There are just natural cycles. With a lot of bugs, with the majority of bugs, they are just R-selected species in general. Most bugs are R-selected. And The second that conditions are favorable for them, there's more rain, a certain plant bloomed in abundance that year, there's a bunch of a plant died and there's an accumulation of organic matter, there was a fire and now all there's more sunlight hitting the ground. Something can change like that, and within just one generation you can go from barely anywhere to just plague proportions of some stuff. So um, over, and it's probably especially in tropical places where things have the opportunity to grow year round, or monsoonal where they have 6 months to just go crazy. um, so he had brought up that people are always concerned about these things, but at the end of the day, and this is my little add-on to what he told me, the biggest threat to any species is not overcollection, it's not that, it is habitat destruction, and basically just that. And I guess major terraforming of a habitat, which is similar to habitat destruction by an invasive species. So as long as conditions are stable at a site and you don't have a condo complex being built, and you don't have an invasive species coming in that is directly competing with that species 24/7, 365, like a lot of fish species and mollusks with in Michigan with crayfish and carp. As long as you don't have that, things are going to go on natural cycles, and when things are in abundance, things take advantage of that naturally. When things are scarce, it's harder to do that. So that's my opinion, is the worst thing that can possibly happen is not over collection. It's very difficult for humans who are not just strip mining things to over collect. It is destruction of the habitat and the processes that enable a species to recover and to adapt. So isopod communities, like 50% people misadvertising and swindling. So that's the other thing with isopods is, we had stuff. I received Cubaris murina papaya. I received the first individuals from Smugbug, and she gave them to me to take home and to isolate. I isolated them, sent some back. Everybody was on the same page. They are Cubaris murina. Papaya is the name of this strain. And within a couple of years, then they started being called Cubaris species papaya. You purposely deleted information that was available for years to call them something else to bump sales. Now you take that with something as obvious as Murina papaya, which I'm sure everybody knows. I'm sure that there's nobody who's been keeping isopods the past 7-8 years who looked at species papaya and said, "Huh, it must be a different species," and not this thing that's been common for, what's it going on, like 6 years now. Um, but new people will. And then when they are sold to larger distributors, which is something that happens now, they will be relamed and mislabeled and all this other stuff. And all of a sudden, it's a giant fiasco. And then you don't even know if you're getting what you paid for because it's species papaya. Is it even Murina? Maybe they mixed it with some other mutations, and all of a sudden you've got a kaleidoscope of things in this thing that you thought was going to be one specific characteristic. So, um, somebody's saying, is it a morph or a locale? That's something that, as you guys saw in my labels, I'm distinguishing, because we do currently, in my sales and this other stuff, the locale information and the strain or cultivar is then listed directly after here. So this would, if this had a morph such as white eye, it would probably be Aranavega pacha. But here's the thing is, when you start getting multiple lines of the same characteristic going from different places, here's where I feel you need a different name to separate the fact that this is not the same as the other thing. So people were doing this with, they were having big discussions about, well, British Columbia, we'll call it Aniscus ocellus British Columbia BC orange and blah, blah, blah. Okay, there's going to be more oranges coming from more different places. You need to add more information to distinguish this from other Aniscus ocellus. Call it something else. Call it tangerine, call it citrus, call it sunspot, call it something else, and then preserving that this was isolated from this locality and it is this mutation. Because the same mutation or morph, which is people have swapped morph and locality and this other stuff very willy-nilly, which is why I go with the current system I have with the locality / strain, and then the locality and the strain or cultivar variety is two separate boxes there. Morphs were like, oh, albino corn snakes are my favorite morph. Okay. Albinism is a simple recessive trait. It is one allele on one locus that causes albinism. And somebody correct me if I'm wrong. This is my understanding of albinism in basically all vertebrates. Is one simple recessive mutation on one locus. It is a variation of a single gene. So when you say a morph, you may look at it and say, wow, that's so different than a wild corn snake. But genetically, it's not. It's one different allele on one locus that is causing this major difference between one individual and another of the same locality. So when you call things morphs, you're kind of just referring to just this one mutation. Then people will keep layering things up with a morph. Then you have like an anarene mixed with an albino, and people call the result of that a morph and all this other stuff. But with cultivar / strain, there's more information there than just saying the mutation or the morph, etc. There is more background information that is carried by having a different strain or cultivar name than just having, oh, it's an albino, which is what made me very upset, because you get the magic potion scenario. The mutation that people are seeing in magic potions is Dalmatian. We have called it Dalmatian. That's what it is, the name for this single locus mutation of, let's just say it's little d instead of big D being wild type. So people looked at that, and all of a sudden, because of the sales point of magic potion, the name became instantly recognizable, familiar to everybody. Oh, I want that. Those are so cool. Blah blah blah. People switched from calling the basal and trackable lines of isopods that express the Dalmatian mutation from just calling them Dalmatian. So you wild collect a new species and you get a Dalmatian mutant in there. The first line that has that is then propagated, you can call it Dalmatian. Then you save the information that's associated with that and say, okay, this line is from this place, blah, 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 blah. Then they started thinking magic potion was the name of that mutation, which is not true, which then led to other problems. Now we have depressum magic potion. I'm sure there's other species that have been isolated a magic potion mutation, but that was never it. Again, the information with the magic potion was that here's all the information, here's everything Jay Fiore and I figured out. There's more than just the Dalmatianism going on here. It's from this location, blah, 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 all this stuff. I'm going to take a sip. And here's the thing. We can sit and we could label everything British Columbia orange, Livonia, Michigan orange, Boston, Massachusetts orange, etc. You can do that, but then the problem that is going to happen with too much of that information up front is it will be cut off, it will be forgotten, it will be confused. People say, well, they're both orange. What's the difference between the two of them, etc. And this is evident because in the history of humanity, this is what happened with every other organism that we kept, short of plants. Plants have been more tracked now, but I think that's more because big horticulture has their tendrils and everything, and if somebody sells something that's patented that they don't like, you're gonna find out about it really quickly. So it's important for them to keep that information. But even with plants, they will have the strain name that is used internally, and then the sales name, which will be different, which I think is ridiculous. Um, Anyways, so what did we do in these situations in the past when you had an organism that was from somewhere that had its unique from that place characteristics that then may have carried other characteristics from that place? You would give it a breed name, like with dogs or with chickens. There is a bunch of information that is then compartmentalized in that name that you can then look up further, and then you can find out, oh yes, the white leghorns had the dominant white gene in chickens, and they were found in this area, and there's all this other information associated with the breed of leghorn, and so on and so forth. So, and then you can also find out that, oh, if I have um, barred leghorns, I don't actually have leghorn proper. I don't have the leghorns that were originally from Italy, which are the mostly white birds with these characteristics. I have somebody who took leghorns from there and then crossed them with barred rocks or plymouths or whatever, and then rebred in the physical characteristics of a leghorn to get the color of the barred rocks, etc., on a leghorn looking bird, etc. So, let's see. What's your opinion on unethical collecting? There is a Drosophila hydi that is just in love with me right now, everybody. Um, uh, do you think overcollection could affect the genetic diversity of wild pops, making them less adaptable? Says Circa. 100%, and we have direct evidence of this. There is I think, some orchid in China, somewhere in Asia, that is collected for traditional medicine or something. It's just collected. That plant is now rapidly evolving flowers that are colored like the rocks around it to avoid the predation from the humans collecting the colorful flowered individuals. So here you get into this complex thing of, how much are we as humans allowed to affect the world around us, knowing that what we do affects the world around us? Where is our responsibility? Where is the line drawn there? And I don't have a solid answer for that question. So, those collectors of that orchid or that flower are 100% altering that population, perhaps forever. But then again, the plant is also adapting to a new selective pressure. So it is adapting to its environment. There's a fitness benefit to these characteristics. And so, it's difficult. So, with some other things, if you go out and you collect, I'm only going to collect the blue individuals because that's the only ones people sell for or buy. Then you sell them as that locality, and it's like, but you're not really giving people, if you sell it as that locality, you're not really giving people a representation of what you found there, And on the flip side of this, captivity is its own filter. For example, my Armadillium vulgare St. Lucia, when I first received them, there was an abundance of different colors in the colony, and they were received very early on in the stages of them being brought into the U.S. So there is a almost certain likelihood that those colors, because all these different morphs and stuff and strains, whatever, were not being as swapped around and carelessly mass produced back then. There's a It's almost certain that those colors, the oranges, the yellows, all that, were endemic to that population of St. Lucias. So that's how the wild ones were. So now my St. Lucias, for a ton of reasons, have undergone tons of genetic bottlenecks in the time that I've had them, because I know that adding from an outside source to them, I will add in genetics from somewhere in the US, from somewhere anywhere in the world. There's so many vulgari out there, and so many breeders have just mixed them, and they use unsterilized leaf litter and all this other stuff. So it is more important to me to preserve the core of the St. Lucia genetics than it is to Oh, I gotta outbreed. Oh, I don't have. There's no more oranges in here anymore. I gotta get that genes back in here. That is not what I'm gonna do. That's not what I've ever been about. So, preserving this core of pure St. Lucia genetics is more important to me than getting that phenotypic diversity back at the risk of compromising the stock with non-St. Lucia individuals, which I would say is guaranteed at this point. So, when I sell them, I will still sell them as St. Lucia's because that's what they are. But I will in their description say, due to various genetic bottlenecks, this group is no longer as phenotypically diverse as it was when I originally received it. And I think that's a good thing to just sort of put out there and to add, because there are traits in those St. Lucia's that, when mixed with other isopods, you get very different things. These yellow individuals that St. Lucia's throw are multi-gene yellows, and I work them into other projects, and they bring yellow like you see out of nothing in U.S. stock lines alone. And so I would like to keep that around in case I need to use that St. Lucia stuff without other stuff interfering in the future. So, John Doe saying, I have albino anemone on my slate, but I think someone has some morph name they gave them already, but I don't know what it is. And then again, you don't know. With the Murina, it's getting weirder because they were brought in from Southeast Asia as well. And then maybe around 2018, late 2017, early 2018, people in Florida isopod people in Florida started saying, oh, I find those in the wild. I'm going to start breeding them. And then they got colonies, but they didn't record where they came from. And the original Murinas were all collected on Bahia Honda in Florida. All of those were collected by me, the original Cubaris Murina stock, from which papaya comes from, was all collected by yours truly from one like desiccated log on Bahia Honda, and maybe a couple from behind like the ranger station or the outhouse building in the leaf litter. So that's where all that stock came from. And now you've got it being mixed with Southeast Asian stock that seems to have a lot more genetic characteristics, or a lot more of these mutations seem to pop up in it. You've got the glaciers, which is whiteout genetics. You've got there's oranges, there's the anemones, which I don't think is Dalmatian. I think it's something else weird. So mixing all this stuff together, you don't know exactly what you're going to get, what's going to come out of it. Duke and Enzo the Iggy's. Here's something that I have found over years of working with isopods. Piebald is almost non-existent, to the point where if you see somebody posting about a pied line, it's probably not something you want to really get into, because piebaldism seems to be a heritable, non-genetic trait. It seems to be might be caused by a pathogen. It might be caused by something else going on. But the biggest example is koi was something that was spread around a lot, and some places still cling to their koi lines that they have for sale as if it were something special. Koi were called koi because they had the modeling of the white and the various other colors of the scaber, and everybody went nuts for them. And then there's orange koi. But with the original koi, the longer you keep them, the more people realize that they don't prove out. You get all these wild types and these other mixtures. Alan Gross used a piebald that popped up in one of his colonies, crossed with a bunch of other stock. Lottery Ticket was his basal stock for selecting mutations from, and it was just a cesspool of different genetics. There's nothing wrong with that for selecting different traits. It's a great way to bring out a bunch of different phenotypes, but the problem is then you get a bunch of things that are hets. You mix a bunch of different strains, you mix a bunch of different pathogens. You do all this stuff uncontrollably. You don't know exactly what you're pulling and how long you need to keep it separated to get it to prove out. So koi, you look at people's koi now, it's like, why is it so special? Oh, I don't know. That's what it is. I got them from some guy at a reptile show a couple of years ago, and they breed great. But now you've lost all the thing that was originally there because it's not a consistent trait. I have piebald scaber from Valerie, who found them on the side of her barn, and I've been breeding them since like late 2017, always selecting the highest expression individuals, and I will still get low expression sometimes. And on top of that, the immatures don't start showing their piebald coloration until they're about 5-6 millimeters. If it were genetic, I would expect to see it that early on, like you see with whiteout, like you see with orange, etc. So when you see people posting piebalds, I immediately think, oh, this probably isn't going to prove out. Even the Silysticus convexus Ukraine pied is not a piebald. They're Dalmatians. That's the Dalmatian mutation, but somebody jumped the gun and called them piebald for some reason. Um, so there was the piebald levis, the Cali mix stuff, which came from somebody's manure pile initially, I think. Tried for a long time to get those to prove out, but the piebald-looking individuals never produced any offspring when pulled, which led me to believe that Cali mix and the piebalds in those levis were affected with some sort of pathogen. Something was wrong with them that made them unable to reproduce. But if you have a colony of them going, and there's some wild types, and there's some high expression and some low expression, you have a diversity of individuals, and you're always producing some that are susceptible to whatever is causing that piebald phenotype. So you always end up having piebalds in that enclosure. But that doesn't mean it's genetic. That just means you have a population that is resilient enough to keep going, but still be infected or affected by whatever is causing it. So Dalmatian, though, is body's white. There's some fleckling of black on it. Could be big chunks, could be small chunks. Some of it's genetic, some of it isn't. And that's the difference. Dalmatian is a simple recessive mutation. I've always referred to it as little d in terms of doing Punnett squares. What gives someone the right to name a locality or morph, says Insect Central? Well, you don't really have a right to name a locality. It's just where you found it. I feel you have some degree of responsibility to present as much information about where you found it as possible without going too overboard, I suppose. Some people do keep it to like, this is the exact rock I found it under. And I think that is valuable. I think that's valuable. I think that the old standard for labeling insect collections was using like a nearby landmark. That's been kind of the standard for hundreds of years. So using a city, using a nearby landmark are tried and true ways of keeping track of this information. So I feel like it is a bit much to go too far beyond that. So for a morph, though, here's the thing. Back in 2018, 2019, when I was still on Facebook and arguing with people about all this stuff, they wanted to have this weird commission, this weird council to like, well, once you've gotten it proven out, and then you can bring it forward, and then you can name your morph. No, anybody can name anything. You could go out in your backyard and collect orange Nizatum and name the line. The only thing is, don't name it the name of an existing line. And when you distribute that colony, share the other information with it. Oh, yes, these are my Nizatum Rumpelstiltskin from my backyard in Cumming, Georgia. And I think they're simple recessive orange. And here they are. And here's what I call them. Great. This is wonderful. We could have a million different strains of one species with one mutation from all over the US, from all over the world, all with different names and all with this information available somewhere. And I think that's great. And I think everybody has the right to do that. I don't think anybody should be able to step in and say, you can't name that. You didn't do this. Even something unproven out. Even if you say, oh, these are just from my backyard in California, and there was a couple of weird, like spotty looking ones in there, and I collected those and I just let those breed. Now I got them going and I want to call them this. I think anybody has the right to do that. And I think anybody who tries to enforce that you can't do that is somebody who feels threatened by what you're producing, who doesn't want to deal with this extra information because they don't really care about any of this stuff. Um, so what gives someone the right? You naturally have it as a human being. Human beings have been propagating and doing all this stuff in captivity for thousands of years. It's why we have dogs and cats and cows and goats and chickens and ducks and a billion other things. And nobody went to anybody and said, hey, you can't take those ducks that you've been selecting for this characteristic. You can't call them that. Um, as long as you're not intruding on somebody else's thing by saying like, oh, mine are actually that. Hey, we've had this line going for 100 years, and you're saying yours are those? Yeah, but no, I said mine are mine because I said so. It's like playground BS going on there. Provide information about what you've produced. Explain what's going on. And if people like it, if they look at it and they say, yeah, I want to keep that. Those are cool. I like them. So what if they're not all this color or that color? I like it like that. They will gain popularity over time. More people will keep them. More people will want them. More people will talk about them, etc. So that being said, again, with some stuff, it's like, when somebody is like, oh yeah, here's my orange whatever at the show, my orange Skaber. Why is that one gray? Oh, they're gray sometimes. You just pick those out. That is not showing that you care, that you know anything, that you bothered to track your lines, that you even know what you're selling me, basically, with this stuff. And I'm not going to pay money for that thing that I have no idea, because you have no idea what it is. Sure, buy it. You want something for your bioactive because you just need an isopod to toss in there for your White's tree frog to eat. Okay, buy it. Spend money. Do whatever. But don't try to come into a sphere where people are meticulously tracking things and doing their best to keep localities and strains and cultivars all separated so that we can have nice things, and say that they're wrong. So, I'm going to need another thing. Somebody brought up, was there proof of Elliptorhina javanica and G. portentosa breeding? I have not seen that personally. I have not heard of that personally. And the problem with reports of crossbreeding is people will just make stuff up and they won't know what they're looking at. They will buy a group of roaches and they'll put them in a tank and say, oh yeah, I put these other ones in too at one point, and they must have crossbred because now they're smaller than they used to be. That's what you're basing that on. You saw the average size go down, and you're not even looking at any other characteristic. You're just saying that they're smaller than they were when you put them in there. Color or shape or literally anything else be damned, and you're saying that's your justification for saying they hybridized. So that's my problem with stuff like that, and why it should be thoroughly vetted. I would love to see pictures. Glacier Murina are not from Florida. Citrus Murina were discovered in Florida. Glacier are from, I think, the Philippines. Let me see if Discord will pull up. Yeah, Glacier are actually from somewhere in Asia, and they were collected straight out of a wild population, if I recall correctly. I have to find the communication here. It's somewhere in my private messages, but Easy Eddie Isopods knows the information for those. So yeah, so again, this is more information that is not readily available. You'll go to an isopod website and they'll be like, oh, this is a cool species and it likes this, and this. Okay, but like what other information is there? Like, even if you just speculate, like, I talked to the guy I smuggled them in from, and he said they found them here or there. Where it's still more information. Exotic Willie says, might be off topic, but while on the topic of localities, I've noticed when researching native isopods, I found it annoying finding information on the location where they were initially discovered. Although maybe I'm just bad at locating that information. Yeah, a lot of that will be in publications of the description and things like that sometimes. Insect Central bring up my histers are smaller now, and it must be because they crossbred. Sometimes yes, sometimes no. It just depends. Captive invertebrates can be much larger than wild ones, and wild ones can be much larger than captive ones. There's a bunch of factors that go in there. But yes, Duke and Enzo says, "Wow, so many sites say glacier are from Florida." Yeah, if you ask Easy Eddie Isopods, he has the information. I believe it was a population of 8 wild collected whiteouts, and they are actually from somewhere in Asia, and they know the town where they were from. But it's much easier to just make an assumption with, oh, it came from Florida and whatnot. Exactly really says, I see, I think, as Kyle mentioned, there's many factors that play into those characteristics. Oh, he's talking about the yellowish reductaniscus that Insect Central found. Kyle, eventually I fear diapause might be needed for Florida inverts someday. I wouldn't worry about that. So yeah, so that's sort of where we're at right now. So I'm at the point where I had considered slapping on all of my roaches that were like older colonies, that they were like U.S. hobby stock. I don't know if that's necessary unless we get wild localities of them into culture. For example, my Vericosa, my Leucia hermetica Vericosa, I'm gonna start labeling as U.S. hobby stock, and because now we have Venezuela, and then there's another locality that might pop up soon too. I think it's Colombia. So that is a good idea, just to keep things separated. But I'm not sure if I'm going to go through and do that with stuff like my Ergola cappuccina. Maybe I will in another 10, 20 years. I don't know. But just kind of knowing where you got stuff from, seeing if you can trace stuff back as far as you can. Again, I know not everybody has the time to do this, and if you go looking down some of these rabbit holes, you'll end up just kind of crazy. There's what is his name? On the table. Glenn Carlson is a very enthusiastic and quirky individual who was messaging me a lot more last year, who sent me some old strains of Discodalis and Cranifer that he did some very meticulous work on tracking, and we got them basically to a person, but we can't seem to get in touch with that person to find out what community or what university or community college she got the colonies from, which is ultimately, I feel, a good end point for some of this information to have, is even just getting it to, oh, these came from UCR, they came from blah blah blah, so we know that these colonies are related, etc., or aren't related. But he hasn't been able to get in contact with this critical link, and that person, I think, may be out of bug keeping now and may have been for 15 years. So the chances of contacting them or finding this information decrease with time, and eventually it just does get lost to time. And another one of these problems with this stuff is, don't assume something will be around forever. Do not make that assumption. Do not assume that you will be able to find everything out about something forever, or that it will even be around forever. UCR, which is where a good deal of roach colonies, maybe not a good deal, a fair number of roach colonies can be traced to, had a pure hisser line, pure Portentosa line that was preserved at another university. And then now, a couple of years after I acquired those hissers, UCR contacted me saying they needed to restart some of their colonies, and they brought up that the original hisser line for UCR was lost, and they replaced it with regular old store-bought, who knows the origins or if they're hybrids or not, common hissers. So I was like, let me send you some of your guys' old stock back. It's the least that I can do to get it back in your hands and keep it separate from the other stuff. So, so that's the thing, is you would assume, oh, it's University of California, Riverside. They've had bugs for probably since the 50s, at least since the 80s. They've had this line of hissers. They're always going to have them. They'll still be there. Somebody will be able to get in touch with them. But no, you don't know if they're still going to have them in the future. The best thing you can find, if anybody's out there, if you go to a university, a museum, a zoo, a college, a whatever, is finding some old professor or associate or somebody who's been keeping the back room colony of bugs alive for, well, at this point it could be, it won't be 100 years, for 50 years, for whatever, that's just had them there, and they got them in that old filthy aquarium or plastic bin from the 70s, and the bedding's never been changed, and there's bran and a water wick waterer, and they just say, oh yeah, we've had these for blah 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 blah, left them here when he left in 1995, and before that I think he brought them back when he went to blah blah blah. That is the best thing you can find, and one of the most exciting things you can find as a bug keeper in your entire life is those ancient colonies that have never been messed with, that have never been added to, that are still going, that you have just that bit of information to go off of with them. So, I know insect extensions. I know people who left the hobby from how toxic the hobby was years ago. Alan says going bankrupt. See, that's the thing is. Alan says, "Oh yeah, wasn't there a zoo that had compromised sister colonies?" Yeah, that's another thing I was going for. So UCR replaced their old colony with store-bought stuff, and now they've got their old stock back, and hopefully they run with that from now on. But I contacted, I think it was it was some other zoo in Ohio. It wasn't the Cleveland Aquarium. It was the Cleveland Zoo, because I saw some videos from 2011 or 2013 of their hissing cockroach colony, and I looked at it and I said, there's some interesting traits in there that I don't see in other hisser colonies and other Portentosa colonies. So I got in touch with them. I said, hey, I'm a cockroach breeder and blah, 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 and I was interested in your stock. I want to know when they were brought, when the zoo acquired them, where they came from. I was wondering if I could get some stock for conservation purposes, captive conservation purposes. And they said, oh, we added to our stock from a reptile show a couple of years ago, so we don't have those anymore. We added to them. And it's just like, oh no, you assume a zoo has their stuff together. You make that assumption, and it is an assumption. So don't assume that everything will be as it is forever. If you can find one of those old backroom colonies that's still going, better yet if it's got a date on it, better yet if you know where they came from, oh, just divine. You wrap your mitts on that as hard as you can. You beg that professor on your knees if you have to get a starter colony out of there. Um, So yeah, so moving forward, this is a good topic. We're over time, but that's okay. I will wrap this up kind of soonish. But um, Duke and Enzo saying, I mean, just to cut off communications. Why do people disappear out of the hobby? All kinds of stuff happen. This is just. I hate to toot my own horn, but people popping up for a couple of years, being around, doing stuff, is just the norm for a lot of this stuff. There's, for reptiles, for just all kinds of stuff. People get into things, people get out of things, people who were very passionate. There's that one movie, I can't remember what it's called. I've never actually seen the movie, but the guy's like, yeah, I had fished for years. I had tanks all over the place. I had the cichlids, the angelfish. I did all this. I did it 20, 30 years or something like that. And then he's like, and then one day I just woke up and I said, you know what? F fish. And just, he was just over them. So yeah, there's I don't. If anybody knows what movie I'm talking about, please let me know. I kind of want to watch it just for that part of things. But people just one day they wake up, I'm done. I'm tired of cleaning roach bins. I'm tired of feeding a billion crested geckos and talking about how cute and perfect and original each of my little foo-foos is. And then one day you just realize I'm spending way too much money. I don't actually care about this. I don't like this anymore. I'm not enjoying it. And then they just drop off the face of the planet and they're gone forever. Again, not to toot my own horn, but been in this business a long time. We're going on, geez, was it 2005 when I got my first hissing cockroaches? It would have been late 2005, like the dead end of 2005, like in a negative-degree day. My first box of hissing cockroaches came to me from, I think it was hissingroaches.com. Ron Tremper and his leopard geckos also raised roaches, and yeah, they came to me in a flat rate priority mailbox. It was, I think, $30, somewhere between $25 and $30 shipped for 20 adult, near adult portentous of unknown origin. And I received them in a box with just egg carton. They were just loose in the box with just egg carton, no padding, just slapped in there like they were sliding around and all this other stuff when they were in transit. And they arrived on the coldest day of that year. Was like -7 or something out, and I warmed them up and they were all okay. Um, and so yeah, I've been around about 20 years working with roaches. 20 years very soon at the end of this year. And I mean, I've seen plenty of other people. Thought there was a period of time where people thought I was gone too. It was like was it 2018 or 2019 something when Roach Crossing went down for a couple of weeks and people were like, "Oh, it's over." And I mean, before me, there were all kinds of other Roach people. There was James at blabberous.com was infamous right before I got into things. There was what was his name? God, I feel like such an old person. What was his name again? Roachman Willis. It was Roachman Willis was a big guy. There was doubled's.org. Maybe don't Google that nowadays. Doubled's.org, who had all kinds of stuff. I have stock that I can trace back to Dexter to this day. There was Matt Kay, who was around a lot on Roach Forum. There was what's his face? Greg something. Greg something. I can't remember what his business was. Greg something did roaches. There was Cody Will with the Roach Lab. There's a lot of people that have just popped up and came and gone. And um, I think I like roaches too much to just say screw it one day. It could happen. I'm not gonna lie, it could happen, but I don't know. I think I got a pretty good thing going on here. My confidence with just propagating roaches, more and more species, has just gone up dramatically since about 2021. I've been extremely confident with a lot of things, and continued returns on a lot of cultures has just kept my spirit aflame. Um, but yeah, so people will just disappear, and then you don't know. And again, this doesn't just apply to just commercial small hobbyist stuff. This happens with zoos, it happens with universities, all this stuff. It really does take passion or a very something to be very high profile in the public image. And even with plant stuff too, there's all kinds of things. A disease will wipe stuff out, and then you've got like one person who's still growing it in a remote place. You got stuff that goes out of fashion, and then suddenly you can't buy it anywhere. You have to get it for sale online. It's just the way that life is. So um, hello, Double Z. I've seen you. Shout out to you there. Um, Alan says, if I ever sell off my entire collection, assume that I'm getting into raising a bear. um, People wanting to fund Alan's bear purchase. Duke and Enzo saying, I have a need for those Columbo Niscus. Oh, Columbo Niscus. I'm going to be listing those available pretty soon, actually. Exactly what it says. How long would you say after your first sisters it took you for you to get settled into the hobby? Pretty quickly. It was by the end of that summer. So I got them in the winter, and then when the new year, and my first babies popped up in like March. It felt like forever. And it was actually the day of the science and engineering fair at U of M where I checked them before we were leaving in the morning, and I saw babies and I got really excited about that. So then it was by like the middle of summer, they were getting gigantic, and then I had all these roaches, and my plated lizards weren't eating enough of them. And then I was like, oh, I just kind of started looking up that there were other roaches that people were keeping. And I thought, oh, this is so fun. It's so easy, blah, blah, blah. And then that would have been like my fall of my sophomore year of high school was when I really deep dived into keeping roaches. And I was on Roach Forum, I was getting colonies off of there and trading and blah, blah, blah. So yeah, I think by the time Christmas rolled around, I was up to about 20-ish species, I think. So between like August of August of 2005 and January 1st, 2006, I was up to about 20 species. So please bring some Parahermetica or Roshina. We'll see. People are trying with some of those. Kyle, you lead the hobby. We need you. Yeah, again, I feel kind of responsible in this. I dislike the word community. Like, I want to say it, but I dislike it because, like, it's overused and it leads to bad things. I feel like I'm so proud of this community. And it's just like, I don't know, is there a different way I can call us? Just like a group of dudes, with dudes being gender neutral because it is, or something like that. The roach people also does not sound quite as flattering. Well, you guys are wigglers, so at least for the Roach Crossing following. But we are on the precipice of a big step of what's to come for roach keeping. Exotic Willis is hoping to break 70 to 80 this year, currently at around 40 species. We have some off-topic discussion about mismolting flugus. I can tell that people are craving the wiggles to bring up these things, but alas, our time has long since expired here. So I'm hoping that that sort of expressed my position on some things. It's basically, I understand we can't with some of this stuff. What was somebody, even with the thumbnail Magnifica? So these are all obviously very distinct lines of Magnifica. I can't pull up the thumbnail and I click on the stream. But even Martin, who I know is good at keeping track of things, it's like, oh, this came from a seller in China who said they were collected around here, and this and that, and you're at the mercy of whoever you're getting the bugs from to get this information, and I'm really trying to provide all of that information right up front on the future site and for everything that I sell. So there is no confusion. There is consistency. Even when Latin names change, there are new identities or things are described, etc. I'm trying to keep tabs on all that stuff for my own purposes, but also for everybody out there too. Alan says this just occurred to me. I've never added any new bloodlines to any of my isopods. That's the other thing with the two things to take away is come on, everybody. Let's hold everybody to a higher standard. This is not the 1920s where things just came in off a boat and you have no idea where it came from, and they say, oh, this came from East Asia far, and then you find out it was actually from Europe, and then it went to like the Middle East, and then it was sent on a ship that was coming from Asia to the United. This is not that time. You have one of the most strongest pieces of technology available. That's a picture of me and Angela in your pocket. Like you can access more information in a couple of seconds than anybody on this planet could access in that amount of time 20 years ago. Just 20 years ago. You can at any time just whip out your phone and be like, how do I do calculus? What's the air laden mass of a full grown swallow? What's what's the current taxonomic name for this genus of spider? You have it at your fingertips. There are millions of people talking on all kinds of social media about every topic everywhere. You can find this information if you want to when something new comes in. There's like the excuse is these links in the chain of people purposely withholding or obfuscating information, and that's always bad. It's always bad. So, um, Edison, I think you're new to the stream. If you're not, welcome, or if you're not, hi again. Edison saying every time people ask me how to learn stuff, I tell them reach in your pocket, pull out your phone, and look it up. That being said, there is value. There will always be value in dialoguing with somebody. My friend Jules was like, Kyle, just use Chat GPT, just use AI to help you write your new website. And I'm like, how do I do that? Oh, well, you do this and this and this. And Jules is a software programmer, whatever, a software engineer. And it's like, that would be great if I could do that. The problem is when I ask Chat GTP or whatever, hey, what are the humidity requirements, adult size, adult coloration, recommended enclosure size for Aranavega erratica from Amado, Arizona? Is Chat GPT going to give me all this information? It could say, here's what we found, blah, 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 and it could give me a bunch of stuff that's just completely BS, that it has no idea. It's pulling from information about other roaches or something. It could grab something from like one of the few other places that might have some of that information up, like TJ's website. But then I would look at it and say, well, I think that this is TJ. I'm not dissing you. You know I love you, but I could look at something that TJ recommends and say, this is crap. I would never tell anybody to keep this species like this. This is how you kill them. Blah blah blah. Like that information has to come from me. If I ask, how would you recommend keeping Nocticola species Vienna Zoo, and where did they come from, and who do they come from, and blah blah blah. And what would you recommend feeding them to? That information just doesn't exist. How can it pull and synthesize information that has not ever been put out there before? So, ChatGPT is just going to reword your existing site at best. Yeah, so maybe it'd be good with taking all originality and any character that my current website and writing style has and making it friendly to the masses. But that ain't me. That ain't doing what I want to do here. ChatGPT, pet millipedes are easy pets for your terrariums. That's hilarious. See, you get stuff like that. I remember one point they said millipedes like cocoa fiber in ChatGPT. I hear they have the frameworks to plan a future plan to regulate it. Yeah, there's a lot of stuff. It's just kind of the most dangerous thing was, geez, Angela and I looked up something and it said like, yes, you can do this for that. Then the next sentence down said, it is recommended that you don't do this. Blah, blah. Like it was this direct, immediate contradiction. I was like, this is dangerous. There are people who are like using this to like dictate their day to day and make decisions for them, and you could make a potentially fatal decision with some of this stuff. So anyways, what I like to just to finalize here is moving forward. I think I've said this before. The future goal of the goal of Roach Crossing since a certain point. I mentioned this in a live stream before. I'm pretty sure. My job is not to sell bugs. That is not my job. My job is to preserve and label and characterize this captive invertebrate biodiversity. That is my job and the core of Roach Crossing moving forward. In most departments, that is the actual job. I do not go into this company, or do not plan to steward this company saying, oh yes, we're going to sell insects, that is our job. No, because that's how you go down that slope of, well, I could just sell them this, and it's not actually that, or oh, I'm just breeding isopods. I gotta make a lot of isopods. They're just prognosis, like they're all the same. You know, I don't care if they're mixed up. Just pick out the wrong colored ones. Um, that's sort of the path I feel people go down. As I'm an insect seller, is you go down the production and the money chasing path. And yes, and also, as Monkey says, my job is also to run auctions. That is, the auctions are a means to the end of powering the captive invertebrate biodiversity situation. So, Porcelioides prunosis complex, says Circa. That's funny. This is exactly a hobby to go to for. No, you can go to bugs for money. There's ways to do that. To make, you can go and you can mass collect isopods in Southeast Asia. Call them species, um, furry little bear cub, and then certain people will buy trillions of them and whatnot, and people will make T-shirts about them, and they will make all this other stuff, and you can generate a big ol' hype for that and make money off that. You can sell supplies. You could wild collect stuff on mass and sell that as your bread and butter. I mean, there's ethical and there's unethical ways to do that. Um, you could breed tarantulas and wholesale them. That's a good way. There's tons of ways that you can make a good amount of money. In fact, again, people will say, "Oh, don't go to this route." No, this money, you can make money doing anything. Is the bottom line. Um, like there's I just saw a video of the guy who's now mass producing the what, Hymenopus, the not Hymenopus, is it Hymenocyrus? I think it's Hymenocyrus. The African dwarf frogs. They're mass producing those for pet stores. Everybody wants them. Everybody wants a little froggy in their fish tank. It's always been that way. And they have a whole company doing that, and they make lots of money doing that. There's ways to do all this stuff, and if you want to go and do it with the, oh yeah, I want this as a, like, I want to make money, I want to be absolutely rich and loaded, and or whatever. That's not my modus operandi with the bug collection that I currently have. So, anyways, $350 for one of those frogs. No, no, they're not. That's no. I'm trying. Those are the ones you see for like $10. Maybe they're more expensive now. I haven't looked at them in the pet stores. I do know now they're selling the albinos of them, and actual Xenopus clawed frogs are much harder to find. I need to get some more of those in my life. I need to get a couple honkers of Xenopus laevis in my life. I miss those fellows. That's that's one of those things where I feel like people over time will realize they miss Xenopus. That the fact that they're illegal in a couple of states, not withstanding. But Alan, I was looking at that Silurana tropicalis spot. I don't know if it's necessarily worth my time, but I'm considering it. I do wish I could get a little group of Levis going, and not the albino ones. I just want like the wild types, like the big chonky honking wild types. Actually, I just realized I haven't been at the Michigan Frog Farm. I need to maybe do something with that contact when I move. Um, but anyways, everybody, I hope this was informative. It was great to get on here and babble some more. A big thank you, everybody. Give Mr. Chris Snyper a round of applause, for it is on his dime that we have had this live stream tonight. Edison talking about a friend who had a big albino Xenopus. He called it the chicken because it looked like a raw chicken. Hobby African rain frogs. Oh yeah, that's right, the little screamy guys. Again, everybody will want them, and then they'll see that they're difficult to breed or this or that, and then suddenly they won't want them anymore. But anyways, good night, everybody. Thank you for attending, and hopefully I'll get another live stream going soon of something. If not, the next one's going to be the March fundraiser for the Florida collecting trip. So, good night, everybody. May your cultures ever be bountiful. Might not be a wiggle, but I'll just put that out there too.